First up, please welcome Dr. Shunsuke Funakoshi from the Center for iPS Cell Research and Application, Kyoto University. Yes. Please begin whenever you're ready.
Yeah, thank you for the introductions. I'm Shunsuke Funakoshi. We are a pre-launch company from Kyoto University, iPS Cell Research and Applications. So first of all, it's my honor to be here today, and I'd like to appreciate all the advisory members and the organizers for giving me this great opportunity. So today I'd like to talk about our business plan regarding the cardiac drug discovery using the 3D mature and multicellular cardiac tissues. So there is a huge issues in the drug discovery. So that more than 90% of drug in the human clinical trial fail due to the safety or efficacy issues. And one of the major reasons of this is low translatability of animal models. And the FDA also announced this statement recently, so that before the human clinical trials, the animal tests are not necessarily required. So I think we are actually facing the opportunity to replace the animal model to the in vitro human organ models. And I'm a cardiovascular physician, so I'm interested in the cardiac field. So in the cardiac field, I want to try this challenge. And to solve this problem, so that here I want to show our platform. So we can generate any type of cells in the heart from human iPS cells, including the cardiac cells, muscle cells, and the non-cardiac cells, and mix together to make 3D structures. And then these cells are very, very immature cells. So to add by adding the maturation factors, we can generate the mature 3D heart tissues. So we have the two platforms. One is organoid tissues, and the other is engineered heart tissues. When we check the gene expression patterns in the single cell level, so we can observe the many multiple cell types inside the tissues, and this gene expression pattern is very close to the human real heart. And this is our strategies. We have already established an in vitro heart disease model, including the ischemic heart failure and the genetic heart diseases, including the cardiomyopathies. And also, we have access to the clinical patient data, including the biopsy data and the myopathy registry data, such as the omics data. So by combination of the in vitro modeling data and the patient data, we can very efficiently identify the new treatment target, leading to the new drug discovery within the short time frames. And our initial targeted disease is DCM, dilated cardiomyopathies. Here is the market size and the patient number. And in the future, we plan to scale the indication to the broader heart failure patients. So let me introduce the two examples of our drug discovery. So this is the in vitro human heart failure model. So the important thing is that fibrosis and the heart failure phenotype, including the increase in the biomarkers, are observed only in the multicellular and the mature cardiac tissues, not using the conventional ones. Using these models, we combined the human data, biopsy data, and the transcriptome data, and selected the potential targeted genes. And then I selected the druggable genes and validated using our in vitro models. Then finally, we found the drug which can significantly suppress the heart failure phenotype, including the fibrosis. So then we performed the validation study using the mouse model. We checked the two models, the pressure load heart failure model and the DCM model. In both models, we can observe the significant suppression of the fibrosis and the improvement in the heart functions. Then let me talk to the next topic, next compound. So our interest is doxorubicin-induced cardiac damage. So doxorubicin is known to the anti-cancer drug which has severe cardiac damage, but there are no treatments. So we have performed a high-throughput screening model using the mature iPS cell-derived cardiac cells by combination of machine learning system. And we performed a phenotypic screening and then identified several candidates to treat to improve the cardiac structures. Then, using the in vitro engineered heart tissue model, we have identified this drug, which can significantly improve the contractile functions in the in vitro models. Then finally check the validations in the mouse model, both in the induced cardiomyopathy model and the DCM model. So here I want to show the R&D plan. So we have two compounds. So the first one we plan to go directly to the phase 2 trials because it's a repositioning drug. And the second one we are doing the optimization of these small compounds to get the compound IP. And our targeted disease is DCM, as I mentioned. So here I want to show the more longer timeline. So we plan to start our company in a year. And the important thing is, in addition to these compounds, we have a potential to continuously discover the new asset using our in vitro model. And here is a funding plan. So currently we are seeking the seed investment to start the company. So this is our team. So our team is composed of the clinician, including me, and the pharmaceutical company members, and the advisors, and the business persons as well. So I believe our in vitro biological heart model could open the new door to the innovative drug discovery. Again, so we are planning to start the new startups in a year. So we are taking the collaboration with pharmaceutical companies and investment. Thank you for your kind attention. If any questions, I'd be very happy.